In this video lesson, we're going to be prepping ourselves to learn about the stages of mitosis. Uh, in order to understand what happens, we first need to go over some major characters and plots that you're going to see in mitosis. So hopefully you went over your questions and have reviewed some of the vocabulary. So let's move on to our major players. Now, let me go ahead and make this larger so you can see it. Now, with our cell division, we need to go over some cell parts that we didn't really focus on too much with the cell unit. Now, that's because we're going to go over them now. So the nuclear envelope is the part of the cell that usually contains the nucleus. And it's pretty good at that. It holds the nucleus in, it holds the chromosomes in. But during mitosis, it actually breaks down, it breaks apart. And that's to expand the area of the nucleus and allow the chromosomes to move. And so just like if you really were cleaning your room, all of the stuff that you were cleaning just might get spread out all over the place. Well, the nuclear envelope breaks down. Now, the parts of the nuclear envelope don't disappear. They just get broken down into the little bits. And we can't see it under the microscope anymore. So you'll see in this picture up here, you see the nuclear envelope in gray. And as the cell goes through DNA replication, and then as it starts cell division, you'll see that the nuclear envelope is just gone. It'll reform when cell division is done and finished. Now, the other major player that we want to look at would be what we used to call chromatin. Now, chromatin, as we understood it, was simply loose strands of DNA. They are actually the chromosomes that are just nice and loose and unfurled. The cell has access to them. That's kind of an interesting question to think about. Why does the cell need access to DNA? We'll answer that in the next unit. But chromatin for now would be all of the strands of chromosomes nice and open. But that also means it's very delicate and very loose. We need to tighten up the ship and get everything nice and ready. So what the cells do is they will take their chromatin and they will coil it around, and I mark these as little black dots. They will coil the chromosomes around proteins. And these proteins are called histones. And it's like spools of thread, and you're winding up the spool of thread, and you make it nice and thick and shorter. They call it condensing because it's making it shorter. It's condensing. Answer the definition of the word there. That didn't help. So here we have, let's practice our terms. We have two chromosomes. One blue chromosome and one red chromosome. Now they're nice and uncondensed. What we're going to do is start wrapping them around the histone proteins. Now you'll notice before we do that, I show them duplicating. Now let's practice our terms here. We still have one chromosome that's duplicated here on the left, the blue ones, and then we have a second chromosome that's duplicated on the right, the red and orange one. Now we're going to organize them. What they're going to do is coil up, and you'll see them get really recognizable as a X shape, right? And so we start to see them coil up and coil up. And they're still tied together. We can't lose sight of that. They're still tethered. And as they coil up and coil up, they start to look like our typical X-shaped chromosome. Now, in this last frame, I'm going to ask you to pause and answer the question, how many chromosomes do we have in this last picture And if you paused, you should know, and you should have said that we have two chromosomes. One here, the blue one, it's duplicated, but it's still one. And then we have another chromosome on the right, the one that I kind of made orange and red. So that's the second chromosome. It's duplicated, but it's still just one chromosome. So we've got two chromosomes. Now, each copy is called a chromatid. So this blue one has two chromatids, one dark blue, one light blue. And then this other chromosome also has two chromatids, sister chromatids, the orange and then the red. And I often get students asking me at this point, why is everything a female term, sister and daughter? Good question. Don't know that. Never could find anything that explained why. We don't have brother chromatids, we have sister chromatids, and then we have daughter cells. So the DNA, the chromatin, starts coiling up and making the chromosomes good and visible. Now, the other thing we need would be a way to move these chromosomes about. And here we get spindle fibers. Now, this is another word that derived from an old use of a term that we never use ever. How many times have you ever heard spindle? Never, right? If you lived in the 1850s and 1880s, a spindle would be far more familiar to you. A spindle was an elongated structure that they used to use in a loom to weave fabric. And so when scientists first saw these structures in the microscope, they kind of said, whoa, it kind of looks like a spindle fiber, and the name kind of stuck, and we've never really gotten out of it. The spindle fibers are long proteins that are made of microtubules. That's a horrible typo. Fix that. Made of microtubule proteins, long filamentous proteins. They're made by the centrioles, and you can see in my two GIFs here, or GIFs, the centrioles start out on the sides, and then they move apart. And as they move apart, they're weaving and making these spindle fibers. And they're going to be connecting to the chromosomes and moving the chromosomes about. So you see the spindle fibers growing and becoming very, very elaborate, and then they'll disappear as well. And they are the organizers of the cell division. They will tie to the chromosomes. And again, you know what? This is a good point. You guys can pause and take a look at this cell in the top. How many chromosomes does this top cell have? And you should agree that this top cell has 6 chromosomes to it. And then this bottom one, again, you guys can pause it and notice this bottom cell should only have 4 chromosomes. And then of course they separate and divide. But let's wait just a sec. Right here, you should be able to see that they condense and make 1, 2, 3, 4 chromosomes. So what we're going to see in the microscope, it kind of looks like these little furry hair. Each one of those little red pieces of fur would be the spindle fibers, and they're what are pulling the dark purple chromosomes. So chromatin, loose strands of chromosomes that condense and tighten up around proteins to make the chromosomes visible. Spindle fibers, the long filamentous proteins that tether the chromosomes and move them about. They grow at the beginning and then they disappear once cell division is over. So what we're going to see here, and let me go ahead and get out of present mode here. What we're going to see here are the stages of mitosis. And let's just go ahead and introduce them, and then we'll be done with this video. We have interphase, which means the cell is not dividing at all. Remember, interphase is simply where the cell is doing normal cell functions. Then we have prophase, and you can move your tile. Interphase, prophase, and we'll go over these each individually. And then we have anaphase. Oh, sorry, I missed one. And let me go ahead and make one real quickly. Why did I miss that? I caught it, thankfully. So we're missing one here, and you will have it fixed by the time you get your slides. Here's our metaphase. Metaphase is the middle one. There we go. Now we have anaphase, and then telophase, and then our daughter cells. So notice we started with one cell, we're ending with two identical cells. Remember, mitosis makes mitose. And so in the process, a very, very simplistic version, we'll go over it in detail, but in a very simplistic way, you can see we're going to line our chromosomes up, divide them, sort them into the two daughter cells, and then we get the two. Now, why we have this weird cell here, they're showing a plant cell. So a plant cell ends up having to build a cell wall here. Animal cell just divides the cytoplasm here. So that is your nice introduction to mitosis and the major players that you're going to see throughout. So I hope that helps, and we will continue in our next lesson.